Chapter 11 is all about gene expression. This first section is just about gene expression in prokaryotes. So the sections further on will talk about gene expression in eukaryotes. So obviously there's a big difference between prokaryotes and eukaryotes, right? So with prokaryotic regulation, just some basic terminology that's going to help are all of these words right here. So a gene, what's a gene found on? DNA. Okay, so your genes are in your DNA. Gene regulation or gene expression is how you control which genes are turned on, so therefore which proteins are made. Okay, so that's the idea of gene regulation and gene expression. That works because of central dogma. So someone remind me what central dogma is.
DNA to RNA to protein.
Yeah, because I totally didn't forget, just wanted you to be reminded of it. So, okay, DNA, RNA, protein. So a bunch of different terms, and part of the problem with these is that they either sound similar or might be hard to keep straight or keep track of the differences between each other. So there's the promoter versus the operator versus an operon. Then there's also regulatory gene versus repressor, and the repressors can be active or inactive, so active or not active. Okay. So stuff's defined here, and there's an explanation of the lac operon here. I'm just going to go to showing you the pictures because I can talk about the stuff from the pictures. Okay, so the term promoter should sound familiar. If I say terminator, those two words should sound familiar. We talked about those with transcription. There's sequences on the DNA that help the enzyme that builds mRNA attach. So what enzyme builds mRNA? RNA polymerase. Okay, so that's this thing here. So the promoter is what the RNA polymerase looks for in order to start making the mRNA. Okay, so it's going to bind to the promoter. If the operator, which usually comes after the promoter, has a repressor attached to it, then the RNA polymerase can't make the mRNA. Okay, so the operon is the name of the whole sequence that controls the gene being made. The promoter is what RNA polymerase attaches to. The operator is what the repressor would attach to. Good so far? Okay, what's the repressor do? It stops the mRNA from being made because it attaches to what? The operator. Okay, so if the operator has a repressor attached, you can't make the protein, or you can't make the mRNA, which means you can't make the protein. Okay, so then this section here, the regulatory gene, is what makes the mRNA sequence for the repressor. Okay, so if this sequence gets a mutation in it, then you can't make the repressor, which means you'd never be able to turn off transcription. Okay, so that could potentially be a problem. That's a mutation that does happen sometimes. But when the repressor is made, it can attach to the operator. Okay, if you remove the repressor, then transcription can take place, which means you can make lactase. Still with me? Go. Great question. We're going to have to address that in a minute. If we don't get time today, we'll talk about it next week. Okay. So this is when lactose is absent. So if lactose is absent, then the repressor is attached. You don't make lactase, or bacteria don't make lactase, because remember, this is prokaryotes, not eukaryotes. When lactose is around, then lactose attaches to the repressor, which pulls it off of the operator, which means the polymerase can attach, which means you can have transcription, and then you can have translation, and you can make lactase to digest the lactose. Seems like a kind of involved process. You really just have to remember vocabulary, so what's called what. So when the lactose is present, it turns on expression of the gene. So environmental changes tend to affect what things are made. If something's present in the environment, then being able to digest that will be turned on. If it's not present, you don't want to be able to digest it. So this operon, the lac operon, is an example of an inducible operon because you can turn it on. So when lactose is present, that turns on the expression of the gene to make lactase. Okay, the trip operon is an example of a repressible operon. So this particular, the product from the trip operon is something you don't always want turned on. So it is turned off when tryptophan is around. Tryptophan might sound familiar. It's an amino acid. So if your body has enough tryptophan, or the bacteria has enough tryptophan, then it's not going to make the enzyme required to make more tryptophan. So tryptophan being around, enough of it turns off the machinery to make more of itself. When there's not enough tryptophan around, then the repressor's inactive, so then the cell will make the enzyme needed to make more tryptophan. Okay? So inducible versus repressible, that's the difference between the lac and the trip operons. Okay? Mostly vocabulary. If the pictures make sense, then you should be able to figure out the vocabulary too. Okay?